Welcome back everyone. Today we are going to discuss this very important topic in the vitamin section, that is vitamin B6. Vitamin B6 is a very important discussion in this vitamin topic. Let's get through this. First we are going to discuss on this vitamin B6 is the active form and coenzyme roles. Active form and coenzyme roles. So there are three different forms of B6. First is the alcohol form. It's called pyridoxine. Second is the aldehyde form. Aldehyde form is pyridoxal and aldehyde form. Pyridoxine, alcoholic form. Third one is the pyridoxamine. There are three forms of B6. Alcohol form is the pyridoxine. Pyridoxal form is aldehyde. The third form is pyridoxamine. Next, I'm going to see is the active form. Active form is the PLP, that is pyridoxal pyrophosphate. Very important coenzyme. Pyridoxal pyrophosphate. You come across this enzyme very often a lot. It's a very important coenzyme. In critical reactions in the body. Important coenzyme in the critical reactions of the body. Most important role is in amino acid metabolism. Amino acid metabolism. The most important role is in the amino acid metabolism. So we are going to see some of its roles, how this PLP, why this is considered as one of the most important coenzyme forms. First we are going to see is transamination. Transamination, there are two enzymes, ALT and AST. This ALT stands for alanine amino transferase. AST stands for alanine. AST stands for not alanine. Stands for aspartate amino transferase. ALT, alanine amino transferase, and AST, aspartate amino transferase. Third enzyme, C, converts glyoxylate to glycine. This enzyme which converts this reaction is glyoxylate alanine amino transferase. These three enzymes, ALT, AST and glyoxylate alanine amino transferase, three enzymes requires PLP, pyridoxal pyrophosphate. Pyrophosphate. This is the active form of vitamin B6. Requires PLP as coenzyme. PLP requires coenzyme. So this, as this glyoxylate is involved in this, so this glyoxylate will form oxalate stones in kidneys. Is called oxaluria. It's called oxaluria. So glyoxylate will form oxalate stones in the body called oxaluria. Which coenzyme is involved here? PLP or B6. You've seen this one, transamination. Next we're going to see is, this is very important, amino acid decarboxylation. Amino acid decarboxylation. Decarboxylation also requires PLP as coenzyme. Requires PLP as coenzyme. For example, we are going to see this conversion of histidine to histamine. Histidine to histamine. Another example is 50H tryptophan to 50H tryptamine. Tryptophan to 50H tryptamine. This 50H tryptamine is commonly called serotonin. So serotonin is derived from tryptophan. This 50H tryptophan is converted to tryptamine 50H tryptamine. Then this glutamate converted to GABA. Very important depressing neurotransmitter. So this decarboxylation, all these reactions occur by removal of CO2. Removal of CO2. Removal of CO2. Histidine upon removal of CO2, histamine. 50H tryptophan upon removal of CO2 forms tryptamine, which is serotonin, and glutamate upon removal of CO2 forms this is GABA. All this requires PLP as coenzyme. PLP as coenzyme. Next example we are going to see is of transsulfuration. Transsulfuration. Here we are going to see some examples. For example, see this reaction, homocysteine when combines with serine. So homocysteine and serine are two sulfur containing amino acids. Homocysteine and serine are two sulfur containing amino acids. They combine in presence of this enzyme cystathionin synthase. Cystathionin synthase. Presence of these enzymes forms cystathionin, which is then further converted to homoserine plus cysteine. This cystathionin is further converted to homoserine and cysteine in presence of which enzyme? This important enzyme, cystathioninase. First enzyme cystathionin synthase. This is cystathioninase. Cystathionin is finally converted to homoserine and cysteine. This is the transsulfuration reaction. You see, from homocysteine and serine we got homoserine and cysteine. These two compounds are reversed. These two enzymes requires PLP as coenzyme. That's why I've said this PLP we will come across very often. Further discussions, especially in the amino acid metabolism, this homocysteine, cysteine metabolism, how they are converted to other forms, this transsulfuration reaction, these two enzymes, cystathionine synthase and cystathioninase, requires PLP as coenzyme. These two are sulfur containing amino acids. Here what happens, there is transfer of this SH group. There is this SH group from homocysteine, this original homocysteine, to serine, cysteine to serine, with which this homocysteine combines to serine, leading to serine being converted to cysteine. Serine being converted to cysteine. So serine converted to cysteine. The final products of this transsulfuration reaction. Remember, transfer of SH group from homocysteine to serine. So this is all about the transsulfuration reaction. Next, we are going to see is the all important tryptophan metabolism, perhaps one of the most important metabolisms, amino acid metabolism section. Tryptophan metabolism, PLP is required. How? We are going to see. So we have this compound 3-hydroxykynurenine. 3-hydroxykynurenine. It's an intermediate product in the tryptophan metabolism. 3-hydroxykynurenine, in presence of this enzyme kynureninase. Kynureninase enzyme is converted to 3-hydroxyantranilate. 3-hydroxyantranilate or antranilic acid. 3-hydroxykynurenine, in presence of kynureninase, 3-hydroxyantranilate or antranilic acid. This requires PLP, pyridoxal pyrophosphate, or simple we can also say pyridoxal phosphate. Commonly don't say pyrophosphate, also say pyridoxal phosphate, because kynureninase requires pyridoxal phosphate as coenzyme. Now this antranilic acid has two fates. It goes for a ketogenic fate. The other one is NAD plus synthesis. This 3-hydroxyantranilate or antranilic acid has two fates. It goes for ketogenic fate and or NAD plus synthesis. Now we have a vitamin B6 deficiency here. B6 deficiency. So this active form PLP will not be enough for this action of kynureninase. To see PLP deficiency, so this kynureninase enzyme will not be properly functioning, which lead to go for xanthurenic acid formation. Xanthurenic acid formation. Why? Due to PLP deficiency. This PLP deficiency can also lead to pellagra. Why? As it is involved in formation of niacin. We have come full circle. In the vitamin B3 or niacin video, we have seen this vitamin B3 is derived from tryptophan. So as this PLP is involved in this tryptophan metabolism, this PLP deficiency leads to xanthurenic acid formation. Also can lead to pellagra, or you can also say pellagra like symptoms. Pellagra like symptoms. Why? This PLP also involved in formation of niacin. Niacin is derived from tryptophan amino acid. So all these things are connected. We need to remember and understand properly. So we have discussed this tryptophan metabolism. Next, fifth one we are going to discuss is heme synthesis. Heme synthesis. PLP is also involved in this heme synthesis. How we are going to see? The starting substrates for this heme synthesis are succinyl coenzyme A combines with this glycine, this amino acid glycine combined with succinyl coenzyme A. This enzyme is ALA synthase. This ALA synthase is a very long reaction, multiple steps reaction. There are multiple steps in this. In short, I am saying, just remember, in the presence of this ALA synthase, the succinyl CoA and glycine combines to form ALA. It stands for alpha levulinate. ALA, alpha levulinate. Presence of this enzyme ALA synthase. This ALA synthase requires PLP, pyridoxal phosphate. This ALA synthase requires to form ALA. This upon successive reactions finally forms our heme. Now we are going to see that what happens in this deficiency in case of B6 deficiency. PLP is not aptly available, so this reaction fails. When this intermediate reaction fails, this synthesis of heme fails. This B6 deficiency leading to failure of heme formation. Failure of heme formation. What we know happens after this, it leads to microcytic hypochromic anemia in peripheral smear. Microcytic hypochromic anemia in peripheral smear is seen in which, due to failure of heme formation. Heme compound mostly important required for formation of hemoglobin and development and maturation of RBCs. It's not properly formed. Why? Due to B6 deficiency. Why? This PLP is required for the coenzyme for the function of ALA synthase. This deficiency leads to microcytic hypochromic anemia, where in the peripheral smear. Discuss this heme formation, how PLP acts as the coenzyme. Next, final one we are going to discuss is glycogenolysis. Glycogenolysis. PLP is a coenzyme for the rate limiting enzyme of glycogenolysis. This PLP is the coenzyme for the rate limiting enzyme of glycogenolysis. What is that rate limiting enzyme? It is glycogen phosphorylase. Glycogen phosphorylase. This is the rate limiting enzyme of glycogenolysis. It requires PLP as coenzyme. Requires PLP as coenzyme. This glycogenolysis takes place in liver and muscle. In this word we can see this glycogenolysis. This lysis means breakdown of glycogen. So this breakdown of glycogen, as we know, the final product is the glucose. Glucose is the form of energy that our body uses. This glycogenolysis takes place in liver and muscle. Another important fact we need to remember is 80% of the storage of pyridoxal phosphate PLP is in muscles. This we need to remember. 80% of the storage of PLP is in the muscles or the skeletal muscles. Glycogenolysis takes place in liver and muscles. We discussed 6 important reactions, biochemical reactions, pathways in which PLP, pyridoxal phosphate, acts as coenzyme. First is this glycogenolysis. Rate limiting enzyme is glycogen phosphorylase. Next is the heme synthesis. This intermediate alpha levolinate ALA synthesis enzyme PLP, whose deficiency leads to microcytic hypochromic anemia in the peripheral smear. Deficiency of B6, microcytic hypochromic anemia. Next, tryptophan metabolism. B6 deficiency leads to xanthurenic acid formation. This is very important. So a lot of star marks, xanthurenic acid formation. This can also lead to pellagra, or more specifically pellagra-like symptoms. We've seen the pellagra-like symptoms. This is the carcinoid syndrome. There is the Hartnupp's disease, and in this PLP deficiency they are involved in the formation of niacin. Niacin is also derived from tryptophan. Next, we have seen this transsulfuration reaction. These 2 important enzymes, cystathionine synthase and cystathioninase. This requires PLP. There is a transfer of this SH group from homocysteine to serine. And so this decarboxylation of amino acids, PLP, this is the coenzyme. Finally, this transamination reaction, most important of all, transamination reaction. Say ALT, AST and glyoxylate alanine aminotransferase. This glyoxylate will form oxalate stones, called oxaluria, if this requires PLP as coenzyme. These are the 6 important phases or reactions taking place in the body which requires PLP as their coenzyme. Next, we are going to see its deficiency manifestations. First is the neurological manifestation. Why? Because it's involved in the GABA metabolism. You know, this GABA is an important depressing neurotransmitter. It's formed from where we have seen above in the decarboxylation, formed from glutamate. Glutamate on decarboxylation form this GABA we have discussed above. So This GABA is an important depressing neurotransmitter. Its neurological findings are peripheral neuropathy, then there are personality changes such as depression and confusion. This confusion and depression leads to personality changes. Person starts to behave weirdly. Then there are convulsions. Then we see there are pellagra or pellagra-like symptoms. Then we also seen this microcytic hypochromic anemia. Pellagra-like symptoms. Neurological findings are due to its GABA metabolism, peripheral neuropathy, confusion and depression, convulsions and peripheral neuropathy. So why these deficiency manifestations are mainly neurological? Why this causes? Because this lipids causes axonal degeneration. Remember this important point. This lipids causes axonal degeneration. This leads to neurological manifestations as is involved in PLP is involved in GABA metabolism. So I've seen this deficiency manifestation, nothing much to see here. Next we are going to discuss is urinary analytes in PLP deficiency. First thing we are going to measure in urine is this homocysteine. We've seen above how this PLP is involved in transsulfuration reaction between homocysteine and serine. So homocysteine is measured. Then xanthurenic acid. We have seen in the above discussion how this tryptophan metabolism, this PLP deficiency leads to xanthurenic acid and the level of oxalates. This is oxaluria. These glyoxalates form oxalate stones leading to oxaluria. The level of oxalates are the urine analytes in PLP deficiency: homocysteine, xanthurenic acid and oxalates. So next we are going to see is the biochemical assay. Simple to remember. First one we see is the erythrocyte transaminase. PLP is involved in the transamination reaction. So this word transaminase in the erythrocytes. So erythrocyte transaminase is analyzed. Next one is tryptophan load test. So tryptophan load test. In this, the suspected patient for diagnosis is given a load of tryptophan. As the PLP is involved in this tryptophan metabolism, this will lead to, if there is a deficiency, this will lead to increase in the level of this xanthurenic acid. You've seen above how xanthurenic acid is formed. Also it can lead to, there is symptoms of pellagra-like symptoms. This tryptophan load test is also very useful. The first major one is erythrocyte transaminase. So these two are the biochemical assays. Next one is going to see if there is some toxicity. Okay, how beta Mbss becomes excess. There is toxicity. Does the toxicity exist? This toxicity manifested in the form of sensory neuropathy. Its toxicity is manifested in the form of sensory neuropathy. The toxicity of B6 exists. We've seen before how the toxicity of B1 and B2 doesn't exist, but B3 toxicity exists. Similarly, this B6 toxicity exists, which manifested in the form of sensory neuropathy. Then I've forgotten this one. In this biochemical assay, and I'll add another one. Third one can also be measurement of PLP in blood. This measurement of PLP in blood. We measure the level of the pyridoxal phosphate or the level of any active form of coenzyme of any other vitamins in the urine or in the blood. So this level of PLP in the blood can also be measured in the biochemical assay. So I'm going to see another topic, important topic here under this PLP. Is this PLP and hormone dependent cancer. PLP and hormone dependent cancer. So I'm going to see here this hormone. Every hormone, cellular messengers have a receptor. This hormone combines with its respective receptor to form a complex. Suppose this is the receptor, this is the hormone. Excuse me. This is the hormone receptor complex. So hormone receptor complex has this part. It's called hormone receptor elements or HRE, hormone receptor elements. This hormone receptor complex has a part called hormone receptor elements. This B6, it inhibits the binding HRC, hormone receptor complex, to hormone receptor elements. It inhibits the binding HRC to hormone receptor elements. This is the function of B6 there. So in case of B6 deficiency, there is enhanced binding of hormone receptor complex to hormone receptor elements. There is enhanced binding of hormone receptor complex to hormone receptor elements. We've seen here the hormone receptor elements and the hormone receptor complex. So this leads to the increased action of hormone. There is increased action of hormone called hyperactive hormone. It's called hyperactive hormone. This increased action of hormones called hyperactive hormone. Why this? This is due to enhanced binding of the hormone receptor complex to HRE. Why? It is due to B6 deficiency. Why B6 deficiency? Because B6 inhibits the binding of HRC, binding of HRC to hormone receptor elements, hormone receptor complex to hormone receptor elements. So this is how PLP, pyridoxal phosphate, is involved in the hormone dependent cancer. A small off topic I think needed to discuss here. So this concludes our discussion on vitamin B6, pyridoxal phosphate. We have seen its active form. There are three different types: the alcoholic form, the aldehydic form, and pyridoxamine, and see the 6 reactions: transamination, decarboxylation of amino acids, transsulfuration, tryptophan metabolism, very important, heme synthesis, and glycogenolysis. There are deficiency manifestations which are mainly neurological. Why? Because the lipids causes axonal degeneration. There are also pellagra-like symptoms and macrocytic hypochromic anemia in the peripheral smear. Urinary analytes in the B6 deficiency, if we suspect, we use homocysteine, xanthonic acid and oxalates level are analyzed in the urine. In the biochemical assay, erythrocyte transaminase, tryptophan lot test, and measurement of pyridoxal phosphate in the blood. The toxicity of vitamin B6 exists and manifests in the form of sensory neuropathy. Then we have seen PLP and hormone dependent cancer, how it's acting on development of hyperactive hormone. The last we are going to see is this RDA. RDA is generally 2 to 3 milligrams per day. It increases to 4 to 5 milligrams per day in pregnancy. Pregnancy and lactation, obviously the requirement of vitamins increases. 3 milligrams per day in general normal situation and 4 to 5 milligrams per day in pregnancy. One last thing we are going to see is the important sources. These are yeast, rice polishing, wheat grams, cereals, legumes, oil seeds, green leafy vegetables, etc., etc. So various types of food forms contains B6. We need to have a complete balanced diet of everything to attain is required 2 to 3 milligrams per day of vitamin B6. With this we conclude our discussion. Make sure to revise this, guys, very important topic, PLP. Thanks for watching. I will catch you in the next one. Good luck.